Good evening and welcome to this episode of Dazzle Unlimited. I'm Steve Steinberg, the series host, and I welcome you to tonight's show. I have a very special guest with me, and we're recording this show in Stockholm, Sweden. It's early December 2014 at a major event that is being streamed by the organizers around the world, the Nobel Week Dialogue. It's a full-day session with experts from all over the world, all focusing on the issues involved in aging and what it means for us and for the planet. My special guest is Dr. Eric Kandel. And Dr. Kandel, you are the 2000 Nobel Prize-Winner in Physiology and Medicine, and you're attending this conference, and you're one of the select people at this entire week of Nobel celebrations and the Nobel Awards and this Nobel Week Dialogue event. In 2000, you were awarded the Nobel Prize in Physiology and Medicine for your research on the molecular and cellular mechanisms underlying learning and memory. And this is, I think, is a fascinating field. But before we get into it, what led you to have the interest in pursuing years of research that led you to achieving this monumental award?
It's a fascinating and surprising story. So I'm a Jewish refugee from Vienna, and I was in Vienna in 1938 when Hitler came into Vienna, and the Austrians welcomed him with open arms. It was absolutely shocking. My friends stopped talking to me, and people began to beat up on the Jews.
Yeah.
And I wanted to understand, and I've wanted to understand for the rest of my life, how could people listen to Haydn, Mozart, and Beethoven one day and beat up the Jews in the next? And I thought I would understand this through intellectual history, and I went to Harvard College majoring in intellectual history. But I met a very well-known psychoanalyst by the name of Ernst Kriss, also a Viennese emigre, who said to me, if you want to understand the human mind, you're not going to do it through intellectual history. You've got to do it through psychoanalysis. So I began to read Freud, and I came to the conclusion that he's probably right. Mm-hmm. And I became very interested in psychoanalysis. And I went to medical school with the sole purpose of becoming a psychoanalyst. But in my senior year, there was a 6-month elective period. And I thought even a Park Avenue psychoanalyst should know something about the brain. And I took an elective on the brain, and I found to my astonishment that I loved it. So different than reading textbooks about, brain science or physics or chemistry. Actually doing experiments, planning them, working with your own hands was so satisfying at many levels. So I decided that maybe I should think seriously about making this my profession. And Harry Grinfest, who was the mentor of this lab and a wonderful person, said to me, why don't I nominate you for the NIH when you graduate from medical school? And in my generation, so I graduated medical school in 1956, physicians were being drafted into the service to take care of service personnel and dependents. But the few that were selected for the NIH could use this as an alternative. So I was selected for the NIH, and when I got there, Wade Marshall, the head of the laboratory, let me do whatever I wanted to. And I thought, what's the central question?
But you were already exhibiting or displaying your brilliance then, if he did
Brilliance
that.
is a very generous word. You mean that he let me select my
Yes.
problem? He was a very laissez-faire chief. He let everyone select their own problem.
Oh.
Yes. It's a long story to tell you why. He had a schizophrenic episode early in his career, which is quite fascinating. And after that, he became a very good administrator, but had lost the passion he had for his own science. So he encouraged people to do their own science. And so I thought, what's the central question for psychoanalysis? And it struck me that it's memory. You know, we are who we are because of what we learn to remember. And so many of our later actions are determined by earlier experiences. And certainly this is something that Freud emphasized, the importance of bringing unconscious aspects of memory to consciousness so you have better control of it in your life. So I thought, you know, understanding the biology of memory would be a terrific problem. And Brenda Milner had just studied this patient, HM, in which she had found that the area deep in the brain called the hippocampus was critical for memory storage.
You might want to explain briefly for our viewers, HM, who was that? What was his situation?
HM was a man who, as a young boy, was knocked over by a bicycle, and he sustained bilateral concussions of the medial temporal lobe, which led to seizures. And these seizures for many years were controlled very well, but after a while they became uncontrollable, and the only, since he had seemed to have scar tissue, the only treatment was surgery. So he had the hippocampus removed on both sides. This had never happened before. People had unilateral lesions, but not bilateral lesions. When it was removed on both sides, he was left with a very profound memory loss and amnesia. And a man by the name of Penfield that pioneered this operation. The surgeon who actually did it was a guy called Scoville in New Haven. And so he called Penfield, terribly upset, and Penfield said, I have a colleague, Brenda Milner, a very good psychologist. She's been studying these patients. Why doesn't she come down and we can make something out of this tragedy, can help other patients? So she studied HM and she described this wonderful, in clear way, the memory deficit that he had. So the memory was he could not convert new short-term memory into long-term memory. I had learned in the Grinfus lab how to record from single cells. No one had ever recorded from the hippocampus with single cells. And I recruited a young colleague that I met there to help me. Alden Spencer were the first ones in the world to record from the hippocampus. Everybody at the NIH was ecstatic. They thought we had done the greatest thing ever. Typical of the NIH. Two incompetent people coming here. The intellectual environment brings them to this new level. Boom. And it was wonderful to have these recordings, and we saw that these cells had interesting features, but by and large they were like other nerve cells that had already been recorded from in the spinal cord. So even though we learned a lot about the cells, it taught us nothing about memory. Zero. So we had climbed Mount Everest, but we had no view. And so I began to realize, you know, I'm a bozo. I'm taking this complicated structure that's critical for human memory to study this complicated problem. I should begin walking before I run. I should begin with a very simple form of learning in a very simple animal and try to understand that first before I take the most complex form. Because in those days, one didn't even know what was the nature of the sensory input coming into the hippocampus. So you couldn't try to figure out how learning occurs. In fact, the Nobel Prize this year was given out for people who realized that the hippocampus, in fact, is very complicated. It encodes for space, most importantly, and memory is related to spatial context in an interesting way. So I began to look around for a very simple animal, and I focused in on a marine snail called the plesia that has a very simple nervous system, 20,000 nerve cells. I was able to isolate a simple behavior, a withdrawal reflex, like a withdrawal of a hand from a hot object, and showed that withdrawal reflex could be modified by various forms of learning, habituation, sensitization, classical conditioning. And I could look in the nervous system to see what is the neural circuit for that behavior and how is it modified by learning. And my work showed that the architecture of the behavior, that is the neural circuit of behavior, is quite specified. But what is not specified is the strength of contact between cells, the strength of synaptic connections, the strength of communication between nerve cells. And what learning does is to change how nerve cells communicate with each other. In short-term memory, if you remember a telephone number, you remember something for a short period of time, it's a functional change.
I mean a physically functional change.
Yes, but it's a molecular mediated change.
Okay.
It does not change the anatomy. But if you remember something for long term, if you remember this interview, it's because an anatomical change has occurred in your brain. And that is a result of alterations in gene expression that lead to new proteins that are shipped down to the synapse and lead to the growth of new synaptic connections. So if you remember anything of this discussion, and I don't urge this on you, Steve, but if you remember something, you will walk out of this interview with a different brain that you walked into this interview with.
My God. And at the end of the show, I was going to say, I'll see you next week, but I'm not sure if it's going to be the same me.
Slightly different, modified as so is my memory.
True.
Modified as a result of this interview.
Well, so what was your what was the outcome of that discovery?
Explore new worlds. Read. Visit literacy.gov and let the journey begin.
30 minutes a day of physical activity can improve your health and decrease your risk of heart disease. So start walking for yourself. Start walking with your family. Start walking with your friends. Start a movement in your community. Start walking. Start something. To join the movement, log on to heart.org/start and help start something now.
What was your what was the outcome of that discovery? What came next? What did lead
I
you to?
decided after that I would continue to work with aplysia, but I was ready to move on to the next level. And this was 1980. It had become possible to modify genes in mice, so I could now return to the hippocampus. Now that we knew more about it, people had also studied it and applied genetic techniques to the hippocampus and see that by and large similar processes involved in learning and memory in the hippocampus as were involved in the snail, even though now I was looking at a complex structure and a complex form of memory. Short-term memory is a functional change, and long-term memory is an anatomical change due to alterations in gene expression. In fact, the genes that are involved in switching short-term to long-term memory in the snail for simple learning are exactly the same genes that are involved in your brain and mine for complex.
So we share the same gene.
Absolutely. Now the fascinating thing is, I've recently become interested in age-related memory loss, not a moment too soon, and I found that age-related memory loss can be distinguished from Alzheimer's disease, and it has a different anatomical locus. Alzheimer's disease begins in part of the brain called the entorhinal cortex, the input into the hippocampus. Age-related memory loss, this is work that Scott Small and I have done, actually begins in a region within the hippocampus called the dentate gyrus. And Scott and I have found that there are particular molecules that are involved, and a molecule that is specifically altered in age-related memory loss is related to this transcription factor, CREB, that switches short-term to long-term memory.
CREB is a chemical...
It is a molecule
term.
that's a molecular switch for turning on gene expression. It turns out CREB binds to another protein called the CREB binding protein, and the CREB binding protein calls to a third protein, the fancy name RBAB48. And RBAB48 is what is deficient in the aging human brain and in the aging mouse brain. That is one of the consequences of aging. It decreases in amount. If you restore that in a mouse, you restore age-related memory loss. Whether this works in people, we don't know yet.
Well, that was basically my next question. Can it be restored, and if so, how?
One would hope. It may be possible to restore it in a number of different ways. I've been working recently with a man by the name of Gerard Kasenki, who's made a marvelous discovery. he's found that bone releases a hormone called osteocalcin. And osteocalcin acts on many tissues of the body, including on the brain, and it enhances memory and enhances age-related memory loss. And we've looked together, and these molecules are boosted by osteocalcin.
Does that suggest that the skeletal structure, the skeletal components, are genetically designed to keep the brain in a good function?
Absolutely. And what happens with aging is that the bone mass declines.
Yeah.
When the bone mass declines, you release less osteocalcin. If you do exercise, you prevent the bone mass from declining, you keep the level of osteocalcin higher, and it helps age-related memory loss.
Steve Steinberg here with a program note. You are watching the television series Dazzle Unlimited. You know, the series airs every Wednesday evening at 8:30 p.m. here on Time Warner Cable of New York City, Southern Manhattan, Channel 79 and 1081. Please make a note of it. By the way, if you have comments for us, or if you'd like to advertise on the Dazzle Unlimited television series, contact us. The email address is dazzle@dazzleunlimited.com. Thanks. And now, please stay tuned because there's more to come on tonight's episode of Dazzle Unlimited.
I'm Ed Begley Jr., and I remember what my lungs felt like as a kid growing up and playing outside in polluted air. I'm concerned about air pollution, and I'm fighting for our right to breathe healthy air. That's why I volunteer with the American Lung Association. Too many people breathe too much dirty air on too many days across the country, which sends children and adults to the hospital at alarming rates. Together, we have the power to change that. Imagine how bad our air would be without the American Lung Association. Please support the American Lung Association and its fight for cleaner air. Take steps now to make sure future generations can breathe healthy air. Healthy air is vital to everyone. We all need it to breathe, live, and thrive. And if you're concerned about air pollution, asthma, lung cancer, emphysema, get involved. Contact your nearest American Lung Association at lungusa.org or call 800-586-4872.
In the mouse, we can show if you exercise the mouse, the bone mass increases, the osteocalcin increases, and you improve age-related memory loss.
Fascinating. So does this get into the whole area of what I've heard many of the experts here recommend? Exercise.
Critical.
Regularly exercise.
Critical. Critical. Intellectual exercise, physical exercise, absolutely essential to enjoying your later years.
Well, what about intellectual exercise relating to
I think there is anecdotal evidence that people who stay intellectually involved, socially involved, do better when they age. There are studies that show, for example, if you take a group of 70-year-old women, divide them into two, have one of them be remedial teachers in elementary school for reading, help kids that have reading difficulty, and the other group does nothing. Those that are intellectually involved in the reading exercises for other kids do better 5 years later than those that don't do anything.
Does this mean that intellectual exercise on a regular basis, maybe a continuous basis, except for when someone is asleep or resting, generates biochemical, bioelectrical improvements or even maintenance in the brain?
Absolutely. That's what it suggests. And there are now video games that are being developed because they're so enticing and seductive. People hook onto them very easily. There hasn't been rigorous evidence that these are effective in age-related memory loss, but clearly this is something that is worthy of being explored.
Well, what else have you come to in terms of an understanding of what's related to age-related, well, physical survival and psychological well-being?
Well, social involvement, staying active in a social group is very important. continuing to work. You know, in America, by and large, people are privileged. In academic life, for example, you don't have to retire if you're functioning effectively. In many European countries, people my age no longer have their academic position.
Because they're
they've
forcibly retired.
They reach a certain retirement age, 65 in some places, 70 in others, and they forcibly have to retire. So I think giving people an opportunity to be intellectually active, to be socially involved, is very worthwhile for society, and of course very worthwhile for the individual.
I remember I used to work years ago at CBS News, and sometime after I switched to another network, Walter Cronkite became 65, and they retired him. And I thought, what? He's at the pinnacle of
peak of his career. No. The other thing is, of course, diet, you know, not getting overweight. That is part of physical exercise, but it's independent. So staying physically involved is very involved. And then, you know, checking your blood pressure, checking your blood sugar, making sure that your health is optimal. And if you have any particular disorder of blood pressure or diabetes, that it should be controlled.
Steve Steinberg here with a reminder. You are watching Dazzle Unlimited, the television series that airs every Wednesday evening at 8:30 p.m. here on Channel 79 of this system. Please make a note of the schedule. If you'd like to contact us with suggestions, comments, or to advertise on the series, our email address is dazzle@dazzleunlimited.com. But for now, please stay tuned. There's more to come on this episode of Dazzle Unlimited.
These hands hold tight. These hands love. These hands support. These hands create. These hands save lives. To empower your hands to help make a difference, call the American Heart Association at 877-242-4277 for a CPR class near you. These hands are wise.
So you can't money? That's easy as pie. Brown bag your lunch instead of going out. $6 save times 5 days a week times 10 years is $21,000. That's a lot of lettuce. Small changes today, big bucks tomorrow. Feedthepig.org.
Unfortunately, we have limited time for this interview. Are there any other insights that you would like to convey to our audience about memory, the role of memory, or aging more healthily, or something in this general context?
We've covered the major points.
And do you have a next step in your research, or?
Yes, I'm interested, and we should have another interview on this, on how memory is maintained. How you maintain memory of your first love relationship for the rest of your life. And we've recently uncovered a very interesting self-perpetuating mechanism in the brain, first of the snail, and now we have evidence for it in the mouse, of a protein that is self-perpetuating. That has been described before by Stan Prusner, but the self-perpetuating protein that he and others have discovered kill the self once they get into self-perpetuating mode. We found what we call a functional self-perpetuation, a functional prion, and this is beneficial for the cell, not damaging. So this is a novel mechanism, and we're very fascinated by it and studying it very hard. Next time we meet, we'll discuss that.
Absolutely.
Wonderful.
Dr. Kandel, thank you very much.
Pleasure.
It's a pleasure here.
Really enjoyed this. Thank you.
Thank you. And again, my guest, Dr. Eric Kandel, the year 2000 Nobel Prize-Winner in Physiology and Medicine. Thank you again.
Thank you.
And I'm Steve Steinberg. I haven't won a Nobel Prize yet, but, well.
You're young.
Yeah. Tune in again next week for another Dazzle Unlimited with another special guest, and maybe I will speak more clearly. See you again next week. Have a good week. Good night.
Wonderful.
Thank you.
This is Steve Steinberg speaking. You're watching the television series Dazzle Unlimited. The series airs Wednesday evenings at 8:30 p.m. on channels 79 and 1081 on Time Warner Cable in New York City, Southern Manhattan. Say if you have comments or would like to advertise on Dazzle Unlimited, email us. Our address, dazzle@dazzleunlimited.com. In the meantime, thanks for watching, and I hope to see you again next week when I'll be back with another episode of Dazzle Unlimited.